All right, good morning. So we're gonna be continuing with lecture five today, and lecture five is about the nervous system, and we're primarily focusing on the physiology for now, as in how it works, and then in lecture six we will focus more on the anatomy of the nervous system. We'll learn the names and the structures and various parts of the brain and the spinal cord and so on and so forth. But today we will go through a majority of lecture five, I believe. So let's go through some slides again to remind you where we left off. Okay, we talked about how nervous tissues are separated into neurons. They are the ones that are responsible for receiving signals and transmitting them forward, and they do not regenerate. They don't do mitosis, for example. So we need to protect them with these glial cells, and there are a lot more glial cells in the body than neurons themselves. And here are some examples of those glial cells. Okay, glial cells and neuroglia, they mean the same thing. So we drew the structure of the neurons already, looking at the different parts, the dendrites for receiving signals. Here I'll show you what we drew last class. We have the dendrites receiving the signals. Some of the signals might be green lights, some of them might be red lights, either to stimulate a neuron or to suppress the neuron. And then you will add up all those signals in the cell body and decide whether to pass it on or not. And if you do pass it on, then it will go down the axon, and then it will arrive at the axon terminal where it will pass it on to another neuron. So you can think of it as like, one neuron connected to another neuron, right? And then connected to another neuron. And then the signal will just go from one to another, right? It's like passing on, like a game of hot potato, if you would. And the axons are going to be insulated with these fatty substance called the myelin sheath, and it allows the conduction speed to be faster. Now, we will talk more about this later in a bit, actually. But I believe I told you we have the central nervous system. Central nervous system only has two things in it, brain and the spinal cord, and everything else that's part of the nervous system that is outside of the brain and outside of the spinal cord, it is part of the PNS, peripheral nervous system, okay? And so sometimes you have the same thing in the CNS and in the PNS, but we give them different names. For example, you already know what an axon is, right? This is your neuron, and then you have the long tail. This is an axon. So when you have a bundle of axon like this, like a group of axons all packed together in a bundle, we will refer to them as a nerve if it is outside of this brain and spinal cord. If it's in your PNS, then we say a nerve. Okay, so like you hear people use the expression, "Oh, you struck a nerve." Okay. Nerve is basically everywhere else in the body except for the brain and the spinal cord, and it's a bundle of axon. If you have a bundle of axon in the central nervous system, then we give it a different name. We call it tract. Okay, they're the exact same thing, but because of the location, we give them different names. It's kind of like, Canada and the US. We use different words to refer to the same thing. Okay. Uh, after eating at a restaurant, we say, "Oh, can we have the bill?" Right? And then for the Americans, they say, "Can we have the check?" Right? Or we say we drink pop, and then they say they drink soda. Right? It's the same thing, but different words. Kind of like that. In the PNS, bundle of axon is called nerve. In the CNS, a bundle of axon is called tract. So these axons are covered with myelin, okay, myelin, or sometimes called myelin myelin sheath, and they are for insulation and speed up the conduction. All right. Now, depending on, once again, whether we are in the PNS or CNS, the kind of cells that forms the myelin will be different. In the central nervous system inside the brain, the cell that forms the myelin, it's called oligodendrocyte. Oligo is little dendrocyte, it's like star shape. You already know cyte, cells. Okay, so oligodendrocytes, they form the myelin for neurons in the CNS. Whereas in the PNS, the cells that forms them is called the Schwann cells. Okay, Schwann cells. So both Schwann cells and oligodendrocytes, they are examples of neuroglia. Okay, they're glial cells that supports and protects the nervous system. Okay, so you can see here, oligodendrocytes, it forms all these myelins. Same thing for the PNS, we have the Schwann cells that forms the myelin. Okay. A signal is transmitted from one neuron to another via the, okay, so I haven't told you this yet. There is one neuron right here, and then there's another neuron right here. So when the signal goes from one cell to another, this location here, where they are kind of very close to each other, and the signal is going to jump from one cell to another, we call that the synapse. Synapse. So every time you learn something new, every time you pick up new information, new skills, you will form new synapse in the brain. Okay, new connections, basically. That's um, would allow you to pass information from one cell to another. Okay, we'll talk more about synapse probably later on today. Myelin sheaths within the CNS are formed by, okay, we just learned about this a couple slides ago, and hopefully you remember it is oligodendrocytes, these little star-shaped cells. So let me give you a better idea of, oh, sorry, we did this already, the flow of information in the nervous system. It's this picture that we drew last class. We're touching the hot surface, and then the afferent pathway takes the sensory information to your central nervous system. You process it and determine what to do with it. And then the efferent pathway will send the information back out to an effector, to an organ, to a gland, or to a muscle, right? And then in this case, you will remove your hand. Now, let me give you another example of that. Let's say you are driving and you see a squirrel crossing the road. Okay, so that's sensory information is picked up by your eyes. Your eyes have photoreceptors in them to allow you to see things, right? To be stimulated by light. And sensory neuron will carry that sensory information to your central nervous system. Okay, so again, this is going to be your afferent pathway. Okay, efferent, depending on how you pronounce it. And then you will process that information in the brain in this case. And you're like, oh, no, squirrel is crossing the street. I do not want to run it over. I got to step on the brakes. So then it sends the output, the motor output, through the efferent pathway. And this time, the effector, effector is the final organ or muscle or gland that will execute the motor output. In this case, the calf muscle will step on the brakes and you don't run over the squirrel. So every day we have many of these um, pathways that are being activated in the body, right? As we respond to various stimulation in the environment, right? Everything we see, things we hear, things we taste, right? All of them is going to go through similar pathways. Many of the things we experience, though, does not require us to have an action, even though you still process that information. Okay. Um, So, for example, like, if you are studying at the library, okay, and then people are chatting in the background, but you are very focused on your study, right? So eventually, your brain just filters those sensory input for you, right? You don't even hear people talking anymore in the background, right? And that's an example where, you pick up the senses from your ear, but then your interneuron decides for you that this is not important information to process at the moment. So there's no motor output required. Okay. Let's just, delete that information. We don't need it. Right? So that's the flow of information. Yeah, sleeping in a loud place, that's another example, right? Sleeping in class, all that stuff. All right, we're gonna take another note here and talk about the division of the nervous system. But before that, I want to show you one more picture, okay? This will maybe perhaps give you a better understanding of the central nervous system versus the peripheral nervous system. I'll write the names out for you in a bit, okay? Once again, the central nervous system only contains two things. That's the brain right here, and then your spinal cord. Okay, spinal cord goes all the way down to about your um, upper back, if you would, as small of your back, and then it becomes like a ponytail, actually. Right? It goes like that, and it extends all the way to your lower back. Okay, so these two things makes up your central nervous system. There are nerves, which are once again just bundle of axons, right? Nerves, a bunch of axons in the PNS. So you would have nerves that are coming out directly from the brain. These nerves, okay, they go everywhere. And you would have nerves that comes out directly from your spinal cord. These nerves, they would be part of the PNS. It's originating from the brain and the spinal cord, but is not part of the brain and the spinal cord. And so we call them the PNS. Okay? In terms of PNS, there are 12 pairs of cranial nerves. They come out directly from the brain. Okay? And 31 pairs of spinal nerves. They come out directly from your spinal cord. Okay, so some comes out in the chest region, some comes out from the abdomen region, and some comes out like even further down. Okay, we have 31 pairs of these spinal nerves. Okay, so they are all part of your peripheral nervous system. Now, what do they do? These spinal nerves. Well, the spinal nerves is going to be a combination of sensory neurons and motor neurons. Right? They would take information to and from the central nervous system. Okay, so as an example, if you touch something hot, like I told you last time, let's say this is hot, then the information will go up the sensory neurons. Right? Sensory, the spinal nerve in this case goes all the way up, and then it enters the spinal cord, and then you process the information, and then the motor information will go back down to your hand, and you will remove it. right? So the information goes from the PNS to the CNS and then back out to the PNS. Okay. Now the 12 pair of cranial nerves, they have a lot of functions. Okay, you do not have to know this. Do not have to memorize this slide. Okay, I'm just showing you what they do. You have a lot of, so of the 12 pairs of cranial nerves, once again, these are nerves that comes out directly from the brain as opposed to come out from the spinal cord. You have a bunch of them dedicated for eye movements. Okay, look, 3, 4, and 6 are dedicated for eye movement. Okay, because like our eyes are so important to us to pick up information from the surrounding, right? Okay, if it's blue, it's motor. uh, um, nerves. Okay, it controls movements. If it's red, it's sensory. So over here, again, like you don't have to memorize this line. Right? You have some dedicated for picking information up from the eyes, from smell, from your ear, so on and so forth. Okay, so those are the 12 pairs of cranial nerves. All right, now back to our notes here. Start a new one today. This is gonna be what is it called? It's called the division of the nervous system. So here we go. It's a short note, don't worry. Division of the nervous system. There you go. So, let's start with the big word here, nervous system. And you're probably sick of hearing me saying it by now. There are two main parts to the nervous system. The central nervous system, which is just going to be what and what. Two parts to the central nervous system. Oops. What are they? Too easy, right? You don't even want to answer. If you're thinking of the brain and the spinal cord, you'll be correct. Okay, I'll give you a moment to catch up. I'm just checking to see if people have done their labs yet. It is due today, right? So if you haven't already, please make sure you complete it. Okay, 9 people. All right, let's continue then. Over here, we have the other branch of the nervous system. Oh, someone asked, the lab is 2.5%, not a lot, 2.5%. The test, which is on Friday, is 20%. Okay, so you want to prepare for that. This is peripheral nervous system. Now, we're gonna divide this thing up a couple times. For the peripheral nervous system, it is going to have two divisions, two branches. Okay? And the two branches are the autonomic nervous system. Okay, A and S. Actually, I should probably put the short form here for them as well, PNS and CNS. Okay, so this is the autonomic nervous system, which is used for involuntary stuff. Involuntary processes. Okay, so beating of the heart, changing the size of your pupil, right? Um, Digestion. All these things happening in the background in the body that you don't have to consciously think about. That's governed by the autonomic nervous system. On the other hand, things that you have control over, things that you can do voluntarily, like moving your arms, talking, walking around, those are governed by the somatic nervous system, the SNS. So this would be for voluntary movements and actions. All right. Now, under the autonomic nervous system, there are two opposing divisions. Okay? We call them antagonistic. Okay, I'll write down for you in a bit. We have the sympathetic division, and this is for fight or flight. Okay, if you are in a dangerous situation, if a dog is running towards you, you either fight it or you flight, you run away from it. Then your sympathetic system is going to be most active in that situation. Okay, so when you're stressed or when you're in danger, and sometimes when you are excited or if you are feeling anxious, the sympathetic division is going to be active as well. Think about like, riding a roller coaster. Your sympathetic system is going to be very active. On the other hand, we have the opposite division, and that is called the parasympathetic division. Okay, so this is for rest and digest. Okay, this is like, when you're just chilling at home, kids are in bed, okay, everything's done, you're sitting there relaxing, watching TV and whatnot, then your parasympathetic system is going to be most active. Okay, so the two of them, these guys, they are antagonistic. Antagonistic effects on organs and glands. Okay? For example, when you are activating the sympathetic division, your heart rate is going to go up. Whereas if the parasympathetic system is more active, then your heart rate goes down. So they have opposite effects. Okay? When you are in a dangerous situation, you want to dilate your pupil. You want to be able to see clearly. When you are just like chilling at home, resting, your pupils go smaller. You're getting ready to sleep. Okay? So the two system is like they are both active at any given time, but one is dominating in a certain situation. Okay. So right now, for example, like I assume you're just like sitting at home, maybe attending class. So the division is not particularly active, but it's active enough to keep you awake. And then the parasympathetic system is kind of active too, because like, maybe if you had breakfast, then you got to digest some of your food as well, right? So, it's like a balance, but in certain situations, one will be dominating over the other. Okay. Do we have any questions about the division of your nervous system? All right. Let's take a look at this. So that's what I was trying to tell you. The ANS, the autonomic nervous system, has two subdivisions and they exert opposite antagonistic effects on the same organ. Okay, so you can read about it and see the opposite effects. Another one here is like this. Okay, the sympathetic system is going to inhibit urination. Okay, like and then the parasympathetic system is going to be stimulating urinary um, the urinary tract. Okay, like when you're resting, digesting, sure, it's time to go to the bathroom. But when you're on the run and you're in a stressful situation, when you're like go, go, go all the time, then there's no time for going to the bathroom. Okay, so a lot of these opposite effects. I already talked about this. You can read it yourself. And then this is parasympathetic division, rest and digest. Okay, it's time for do some like housekeeping stuff to maybe promote growth, wound healing, so on and so forth in the body. Okay, let's try some polls and see if I have any luck today with answers. Okay, thank you. So we are choosing everything that's part of the peripheral nervous system. It's quite straightforward. You just have to choose everything except for brain and spinal cord. So this, and this. A, C, and E. Okay. Brain, spinal cords are CNS. Everything else, PNS. How about this one? Hey, just look at the flow chart that we did. I hope you copied it down. Uh, you should be copying the notes as we go to help you understand a little bit more. Okay. The answer is ANS and SNS. Okay. Again, you can't do this from your memory. You have to look at this for now because you just learned it, right? So PNS is separated into ANS and SNS. Okay, that's it. Hey, can you guys tell me like do you guys have lots of tests this week besides mine? I know like mine is on the Friday. Is there like other stuff going on that I should know? Do you guys have chemistry or math or English or whatever? Yeah, tell me what you guys have, please. Two more? In addition to mine, you have two more? Okay, when is it? Math is when?
Yes, sir. Math is tomorrow.
Math is tomorrow, and then what about Cam?
Uh, and English is also coming in, like has lot of writings and then have to review.
Yeah, and
And
chemistry is?
almost every subject.
Yeah. And when's chemistry? Do you
And
guys have chemistry this week?
yes, chemistry is on Monday.
Oh, Monday. Okay.
Yeah.
I mean, I don't know how to like would it help if I extend my I'm not gonna postpone it, but would it help if I make the window longer for the
Yeah.
anatomy
Yes,
for you
sir.
guys? Yeah?
Please, sir.
Okay. How about
Thank
like
you.
yeah, maybe I open it Friday to Sunday. Like I mean for those of you who want to do it on Friday and get it over with, sure. But maybe I open it a little bit more because I can kind of tell people are not really here. Like, either they're studying or if you're in class right now, I don't know if all of you are actually here. I feel like some of you are just like turning it on to make a show and then you go study or something. Okay, yeah, I'll do that. I'll extend it from the deadline to Sunday evening. Okay, but you can still do it on Friday. Okay, I hope that will be a
Thank
little
you
bit
so
of a
much.
reprieve. Yeah, yeah, no problem. Okay, I know it's
It was
tough.
much needed.
Okay, yeah,
Thank
yeah,
you.
yeah, no problem. All right. We do have to continue with the lessons though, since you're already here. And we're gonna take a very long note, okay? This one's gonna take, almost 45 minutes or 1 hour, depending on how it goes. So please, I need you to have two pieces of paper, okay? One piece, we're gonna be writing notes and we're gonna be doing drawings. And then the other one, we're gonna be drawing a big graph. Okay, we will switch between the two back and forth. As I tell you more and more and more, I will start drawing the different parts of the graph, and the two will complement each other. Okay, so get ready. And again, like please, if you are there, you have to copy the notes along with me. That's the whole point of me doing the notes, right? It's much easier for me to just read the PowerPoints to you, but I don't think that is as effective for you than to, write all the notes. Yeah, so have two pages beside each other. I will be switching back and forth between the two. Okay, don't cram them onto the same thing, especially the graph. Okay, do a very nice big graph like the one I'll show you, and that would be a useful cheat sheet for you for the quiz when you eventually have to do it. Okay. So this note um, is called action potential. Action potential. So what does that mean, right? Action potential is basically the mechanism, the method, the mechanism of signal transmission down an axon, okay, of a neuron. Now we have another name for signal. I mean, signal is fine, but sometimes you will see the notes in the PowerPoints or in, things that you read. It's called an impulse. An impulse is essentially an electrical signal. Electrical signal. Okay, so we already know this is our neuron, and this is the axon. And for simplicity, I'm not gonna draw the myelin, okay? But the signal is gonna go down, right? So I'm gonna explain to you how the signal gets passed down. Okay. I was thinking, you guys should talk to my first semester class. They are already complaining it's too much work and it's only week 5. You guys should, tell them it's going to be much more work in second semester. Scare them a little bit. Anyways, so we're gonna focus on the action potential, but first I gotta give you some background information. Background information. There we go. Okay, so the first background information I need to introduce to you is something called the sodium, and have some colors, guys. Okay, you need like 3 colors for this note. Sodium potassium pump. You actually learned this from me last semester, the sodium potassium pump. Okay. Let me just quickly show you, see if it jogs your memory a little bit, maybe, maybe not. I don't know. I was just doing it with my other class the other day, right? When we did this active transport stuff on the protein pump, we talked about sodium potassium pump. Okay. And if you don't remember it, totally fine. We're gonna go through it right now. Okay? So this sodium potassium pump. Sodium potassium pump, it is a type of active transport. Meaning that is requires ATP. Okay? So we have the cell membrane here. The axon is part of the cell, so it's made up of the cell membrane, of course. And you would have the sodium potassium pump right there. Okay, sodium potassium pump. This would be the outside of the cell, and this would be the inside, the cytoplasm. So what the sodium potassium pump does is it uses energy to pump out sodium ions. And it will pump 3 sodium ion out in exchange for 2 potassium ion in. Okay? The pump transports 3 sodium out of the cell in exchange for 2 potassium into the cell. Okay, this is a very complicated process. The note that we are taking. Okay, so please ask questions along the way if I'm not making sense. Okay, so every time it's going to pump 3 sodium out and then it's going to bring 2 potassium in and it's going to use ATP. So if this continues to go on for some time, then you can understand why there is going to be a high concentration of sodium outside and low concentration of sodium inside. Because you keep on pumping it out, right? And that's why you need ATP to keep this going, because you are going against the concentration gradient. Similarly, for the potassium, if you keep on bringing it in, then you will have low potassium outside and high potassium inside. Okay? And once again, you're going against the concentration gradient. Now, I want you to think about this, okay? And like really think about it, because it will set the stage for you to understand the rest of the note. If we do this along the entire cell, okay, so this is the axon, okay? This is the axon. Oops, I just crashed the program. I gotta fix this thing. I don't know how to fix it. Keeps on crashing every class. All right, there we go. Let's try that one more time. It doesn't like me drawing these shapes. There we go. Okay, it works. So this is the axon. This is the axon. Like that axon is like the tail part of the neuron, right? And I'm just zooming in on the axon. And you have the outside here once again, outside, and then this would be the inside, as in the cytoplasm, okay? And then this would be outside again. And so I'm not drawing the phospholipid bilayer, right? This is the membrane and this is also the membrane. And then when it gets to the end, you will have the axon terminal like that, right? Okay, we're not going to draw that, but just so you know what we're looking at. So you would have the sodium potassium pump along the entire axon. So you have it here, you have it here, like this, here and there. And all of them would be pumping sodium out, 3 of them. Yeah, you can put the 3 if you want. And again, use colors if you have. It will definitely make more sense. And then you are bringing in 2 potassium. So as you're drawing this, I want you to think about what's going to happen to the charge of the membrane. Is it more positive outside or is it more positive inside? Remember, you are pumping 3 positive sodium out in exchange for 2 positive potassium in. If you keep on doing this over time, where will it be more positive? Now, I hope the answer is quite obvious. It is the outside, right? Because you get more positive leaving than you are bringing in, right? 3 positive leaves and 2 come in. Essentially, every time you are losing a positive. So over time, the outside is going to be more positive. Okay? Which means the inside is more negative. Okay? So I'm going to write this down for you. The sodium potassium pump causes the inside of the axon to be more negative relative to the outside. this difference in charge is called the I gotta choose a different color. It's called the resting potential. And it is approximately negative 70 millivolts. I'll explain. Just copy first. I'm just looking for a picture to show you. Um, oh, there it is. Okay, so what does this mean? All right, so remember the black lines here are the cell membrane. If you zoom in, if you take this guy and then you zoom in, then it looks like this, right? Okay, so that's just so you are aware of the relationship between all these drawings. And over time, the inside will be more negative than the outside, but exactly how much more negative, right? If you're gonna measure it, then it is approximately negative 70 millivolts. Okay, so what does that mean? Well, if you think of a battery, okay, and I'm sure you probably have access to some battery. You can look at the battery of like a double A battery. It says 1.5 volts, right? So what does that mean? 1.5 volt basically means if you look at the positive terminal and the negative terminal, and on the inside of the battery, I don't know if you know this, but it's like separated by a barrier, right? If you would, and the charges cannot get to each other unless you connect it with a path, right? And that creates a current and that generates electricity, right? So it's the same thing as the cell, as our axon. The inside of the cell membrane is like the negative terminal of a battery, and then the outside, the surrounding, is like a positive terminal of the battery. And the barrier that's separating the charge is essentially the cell membrane. Okay? So how much charge difference are we talking about, right? So we use voltage to describe these charge differences. In a battery, it's 1.5 volts. Okay? For a cell, it's much, much, much smaller. We don't want to like electrocute ourselves, right, so to speak. So we measure it in millivolts. I'm sure you learned this in math, right? Milli is a thousandth of the base unit. So this is negative 70 millivolt. And this is called the resting potential. Okay. Resting, what does resting mean? Resting means the neuron is not active at the moment. Okay. Not all the neurons have to be active all the time. I mean, like, if your neuron is not transmitting any signal, then it's not active, right? Then it's resting. Right? Like I'm not feeling any pain from my finger right now, so there's no pain signal going up. Those neurons are going to be, at rest. And if I measure the difference of charge on the inside versus the outside, the number I get is minus 70 millivolts. And that's an important number to remember. Lots of information so far. Any questions at all? Help me out, guys. Let help me help you understand. Do you have anything at all? Okay, the word potential, let me define that a little bit further for you. Uh, Potential right here. I'm just going to put it as a side note. Potential is basically describing the charge difference. Charge difference across the membrane. At least that's what it means in the context of what we are talking about. Yeah, sure, right? Yeah, someone said 1.5 volt. Is that the resting potential for the battery? Yeah, sure. Right. You know those rectangle ones, right? Those the 9 volt battery, right? That's would be like the resting potential, sort of. Okay. So potential is just a charge difference across the cell membrane. With loosely speaking, the word potential is like the word temperature. Okay. So when we say temperature, I mean, it depends on what you're talking about. Like, are you talking about room temperature? Are you talking about, temperature outside? Are you talking about temperature when your AC is on, right? So temperature is just a word, but the temperature might change depending on your reference point and depending on what's happening, right? Okay. Room temperature is like 20 degrees, right? Okay. Outside temperature today is like minus 15. Right? So like it all depends on what's happening and your reference. And that's the same thing with the word potential. The potential, as you will see, is going to be negative 70 when the neuron is not doing anything, but when it's trying to pass on signals, when it's trying to communicate with other neurons, that number, that minus 70, will actually change. And when it change, we will use different adjective to describe the potential, just like, room temperature or outdoor temperature. Now we're going to say resting potential, threshold potential, action potential, right? So we're going to have different adjective in front of the word potential, but it's all the same. It's all about measuring the charge difference on the inside relative to the outside. Is that okay? Like, am I making it worse? Or do you understand? Let me know, please.
We understand, sir.
Oh, thank you, thank you. Okay. So that's background information number one. Okay, woohoo, done. Let's do background information number two. Background information number two. Okay, so this one, it's called I mean, there are a couple names for it. I guess we'll just call it voltage sensitive gated channels. It's a mouthful right there, and I'll explain. Voltage sensitive gated channels. Now, I hate to keep on bringing back to last semester stuff, but I want to show you to see, hopefully jog your memory a little bit, right? Again, we took this note last semester, and under passive transport, we have something called channel proteins. To refresh your memory, channel proteins are proteins that are in the membrane and they form a tunnel for ions to go through, right? See, it says right here, used by charged particles. Right? Here it says calcium, but other ions can go through the channels. And of course, in the context of what we're talking about, the two major ions that we are focusing on are the potassium ion and the sodium ions, right? So these are channels like the one I showed you, but they have two additional features. Number one, they are gated. Gated means there is a door, there's a gate to the channel. So you can open the gate or you can close the gate. Obviously, if you open the gate, then things can pass through, and if you close it, then nothing gets through, right? So what controls the opening and the closing of the gate? Well, these gates are voltage sensitive. In other words, the gates, the channels will either be open or closed depending on what the potential of your membrane is going to be. Okay? Like if it's at minus 70, then they are going to be closed. If it's at some other numbers, then they will be open. So that's what it means by voltage sensitive gated channels. Okay, so let me write that down for you. These ion channels can open or close depending on the membrane potential. Okay? Like what charge difference are we talking about? Right? If at certain numbers, they're going to open. Right? At certain numbers, they're going to be closed. So there are two kinds of channels we want to talk about. And I'm just going to draw it here to show you. Actually, you know what? Scratch that. Let's do it again properly here. So again, I'm not drawing the fancy membrane, okay? Like I'm just gonna draw a line. It's much easier that way. And I'm gonna put this guy right here. This is going to be our sodium channels, okay? And I'm gonna draw it with two ovals like a door. Sorry, I haven't done this for quite some time, so it's a little bit unorganized. So there we go. That's the membrane and that's the door. And right now this is the closed channels. Closed sodium channel. Now, because of the sodium potassium pump, oh boy. One more time, hold on. Because of the sodium potassium pump, you're gonna have more sodium on the outside than the inside, right? Does that make sense? I think about what I just said. Because of the sodium potassium pump, right? They keep on pumping out the sodium, you're gonna have more sodium outside than inside. So naturally, the sodium wants to come back in, right? Because things naturally goes from high concentration to low concentration. Right? They're like, oh, stop sending us out. It's so crowded here. I won't go back in. But they can't because the sodium channels are closed. Okay? There's no way to go through. They can't go through the membrane because the membrane it blocks charged particles. But they want to go back in. So how do they go back in? Well, the only way to go back in is to wait for our sodium channels to open. Right, so I'm gonna open, like think of a door, right? Opening and closing, right? So this is an opened sodium channel. Okay, so when does it open? Well, opens at negative 60 millivolts, and then it will closes at around positive 30 millivolts. Okay. Now, if you read this stuff online or if you watch some videos online for whatever reason, they might give you different numbers. Okay. I round the numbers to whole numbers to make it easier for you to remember. Okay. So I just don't want you to get confused. So, at resting, it's negative 70. So negative 70, it's lower than negative 60. So it's going to be closed. But when it gets to negative 60, it will open. It will stay open until it gets to 30. Okay, just write these numbers down. Like if you don't fully understand it, like what the number means, don't worry. It will make sense later on. I just need you to write down for now. Now, tell me, once the doors are open, what's going to happen to all the sodium? Right? They're like, oh, doors open. We can finally move. Let's go to the less crowded location. They're going to move inside. Okay? But not before then. Right? You can only move when you open the doors. And obviously, or maybe not obviously, but like, these are sodium channels, so only sodium can go through, right? The potassium, they can't do anything. They cannot go through the red doors, so to speak. They have to wait for their own gates to open. Okay, so similarly, we're gonna draw these things for the potassium. There we go. Potassium right here. Close and open. And where do we have more potassium? More potassium outside or inside? That's right. Because of the sodium potassium pump that keeps on pumping them in, we have lots of potassium inside. They want to go outside, they can't because the doors are closed and they have to wait for the doors to open. As soon as the sodium gates are open, sorry, potassium gates are open, the potassium will leave the cell. Okay, so what's the opening hours, so to speak? Well, they opens at 30 millivolts, and then they will closes at around negative 90 millivolts. Okay, again, like if you can't connect the meaning of the numbers to the events yet, that's okay. I hope it will make sense as I explain more. For now, just understand the doors will open at some number and closes at some number. It's the same as the mall, okay? Mall is going to be open at 11 a.m. It's going to close at, I don't know, 7 p.m. Right? So those are just numbers, right? Between those numbers, you have access to the mall. Outside of those numbers, all doors are going to be shut. That's basically what's going on here. So here, once again, we have the closed what is this guy? Potassium channel, and then this is opened potassium channel. All right. Now just one extra note here. Okay. Let me explain first and then I'll write it down for you, huh? All right, so take a look at my low-tech hand animation here. Okay, so these are the doors, okay? Right now it's closed and then this is open. So here's the thing, right? They're closed, and when you reach the triggering voltage, because they're sensitive to the voltage, right? When you reach the triggering voltage, they will open. And then if the voltage reaches the closing voltage, then they will close. But here's the thing. If you open the gates, if you open the channels and then you shut them close them afterwards, then they enter this period called the refractory period. I'll write it for you, okay? And during that refractory period, the gates are like resetting themselves and they will become non-responsive, right? So if you open it at minus 60 and then it reaches positive 30 and then you close it, and then it's like reconfiguring itself during this time, even if you reach minus 60 again, it will not respond. Okay, so as it's resetting itself, even if you reach the triggering voltage, doesn't matter. It's not going to respond. You're going to give it some time for it to, reset itself. Does that make sense? Okay, so let me write down for you. Once the channel opens and then closes, it will enter a refractory period. where, during which it will become unresponsive. Okay, it's a very short period. It will reset itself, and then when it's ready, it can go again. All right. Now we're gonna take that's all the background information I need you to know. We're gonna take a short break, but any last minute questions before we head for the break? Okay, so this asking the refractory period again. Okay, so the refractory period is the wind. It's like after open and then closes, and then during this time, it will not respond. Even if you reach -60 again, it's not gonna open. You gotta wait for it to reset itself. So during the resetting, when it's not responding, we call that the refractory period. Okay. Do we have an analogy? I can't think of one. Oh, you got it? Okay. All right. Um, yeah. All right. So we'll take a break. We will come back at 9:45. Okay. And we'll do the rest of the note. Okay. We haven't used the second piece of the paper yet. We will uh, when we come back. See you soon. All right. So here we go. Let's continue. I went to the bathroom during the break and then I thought of an analogy to help you understand refractory period. Okay? It's like this. After you go to the bathroom, you flush the toilet, and so the water comes like that, okay? And then you cannot flush it again. Like even if you try to flush it again, there's no water. You gotta wait for it to refill first, right? And then you can flush again. So as it refills, now that time that you have to wait for it to refill, that's called a refractory period. And that's like the gate. Okay, once it open and closes, you gotta wait before you trigger it again. That's essentially what it means. Okay, so let's talk about action potential now, and how it relates to transmitting a signal. Okay, let's do this. It's, a lot of drawings again. Let's start with resting potential. Resting potential. Resting potential right here. Okay, so we're gonna draw this thing. Try to draw it nice and big. Okay, because lots of information will go onto the drawing like that. Okay, with colors, please. We have these little purple ovals. Okay, so we know those are sodium potassium pump. Okay, I'm not gonna draw maybe I'll draw for one of them, okay? Right, it's gonna be pumping 3 sodium out and 2 potassium in for all of them, right? So I'm not gonna do that for everything, but that's true. Okay, maybe let me just give it a little bit more space here. Where is that? There we go. It's a little bit more space. And we're gonna draw more channels. We're gonna have these sodium channels, and because we are sitting at -70 at the moment, all of them will be closed. It doesn't really matter where you put it. I just put them anywhere as you see fit. Okay, some over here. Okay, they don't necessarily have to be right next to each other. They're just there, here and there. Right? So that's going to be closed sodium channels. Another one here, closed potassium channels. And we're gonna draw some dots. Okay, if you don't have colors with you, you can write the word sodium, but it's a lot of words to write. So if you do have colors, then you just do the dots like this. Okay, so sodium outside, more. Let's just maybe leave it at the top. Bottom would be the same thing, okay? And on the inside, not so much, right? And for the potassium, it is going to be the opposite. Okay, so again, if you don't have colors, you will put the word K plus instead of the dots. And you will have some on the outside. Not so much though. Okay? And if you really don't want to do the dots or write the words, you can put high sodium outside and low sodium inside. And it's it's low potassium outside and high potassium inside. As you're drawing this, just make a mental note that the sodium really wants to come back in because it's too crowded outside, and the potassium really wants to leave. Okay? Think of this as like Black Friday or something. Okay? All the customers are lining up outside of the mall. They really want to come in, but all the doors are closed. They have to wait for the opening hours. They have to wait for the opening voltage. Once they open, then they will come in. Okay? And same thing for the blue stuff. The blue are like potassium, they want to leave, but they can't because their channels, their doors are closed. And at the moment, we're going to draw this again. We have negative on the inside and positive on the outside. So I'm going to put a number here that's negative 70 millivolts. And same thing here, negative 70 millivolts. And understand that you don't have to draw this next part, but this is very long, right? It's like the axon. It goes all the way, like really, 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 really long, right? All the way to the axon terminal. Okay. So this is just like, two chunks of it, but it's really long. So keep this with you. I'm gonna start another note, and this would be for the graph. Okay, so this is for the graph. This is action potential graph. Okay, so on a separate piece of paper, I want you to draw a graph that looks like this. I'll switch back and forth, okay? And I'll post both, obviously. So there is the y-axis, and then there is the x-axis. On the y-axis, we have membrane potential. What's the charge difference across the membrane? That's what it means, right? And it's measured in millivolts. And over here, it is time, and it's really quick, milliseconds. Okay, these things happens really fast. Think about it. If you want to move your baby toe, right? Like you can move it like almost instantly, right? So the signal has originated from the brain, it has to travel down the spinal cord all the way to your baby toe, and it's like super fast, right? 100 meters per second, right? Really fast. All right. So on the y-axis, when our neuron is not doing anything, it is sitting at minus 70 right here, minus 70 millivolts, right? And it's not changing. It's not changing. This is called the resting potential. Resting potential. The resting potential is created by the sodium-potassium pump. Okay, maybe I'll not use red here. Sorry. Let's use purple. Okay, so the resting potential is created by the sodium-potassium pump, and at this point, we have low sodium on the inside. It's always on the inside that I'm talking about, okay? Unless otherwise specified, and high potassium. So that's the situation right now. Okay? If the neuron is not triggered, if it stays resting, then it's gonna be at minus 70 like all the time. Nothing's happening, nothing's happening, nothing's happening. Boring, boring. Okay? However, if there is some kind of trigger, right, so we draw like a lightning bolt usually, some kind of trigger. This is a stimulus. Okay? A stimulus, a trigger pushes the membrane potential to minus 60 millivolts. Now I cannot explain to you how it gets from negative 60 to negative 60 just yet. If I explain to you now, it's going to be confusing. Okay, I have to explain a bunch of other stuff first, and then at the end, I'll circle back to this point, and then you will definitely understand how it goes from negative 60, 70 to 60. Okay, just right now, just like take a step of faith and just trust me. There is some kind of trigger that's going to be arriving. Like if you touch a hot surface, that's a trigger that will push the membrane potential from negative 60, sorry, negative 70 to negative 60. And then this is negative 60. This is not to scale, right? The graph doesn't matter. Negative 60. Right? Negative 60 is called the threshold potential. Remember, we have different adjective to describe the potential. Resting potential, not doing anything. Threshold potential is negative 60. Once you cross the threshold, then you're going to fire the neuron. Anything below the threshold, nothing happens. Okay? So it's a line that you cross. Once you cross that, the signal is going to go. There is no coming back. Anything below, nothing happens. Okay? So the signal arrives and it pushes the, oh boy, let me try that one more time. It pushes the membrane potential towards 60, negative 60. What's happening at the threshold of negative 60? Let's go back to the other picture, the other note, and take a look. Remember, we have all these sodium, potassium channels. They're all closed at the moment because it's sitting at minus 70. However, if we change the number to minus 60 because the stimulus is coming, then you can see negative 60 is actually the opening voltage for the sodium channels. Right? Potassium channels will still stay shut, but the sodium channels will open. And when the sodium channels open, right, you should understand that the sodium are going to be coming in. Right? So let's draw that out and see what happens. Okay. Going beyond the threshold potential. Going beyond the threshold potential. Okay, so we're going to start a new drawing. It's not going to be as busy as the other one. I will not be drawing the sodium potassium pump because they don't really do anything important at this moment. Okay, we will have the sodium channels open because we just crossed the threshold, right? I told you a stimulus arrives and then it pushes it over -60. So these channels open. However, further down the axon, it is still sitting at -70. So the doors over here are still closed. They're not opening yet. Okay, because the stimulus, the lightning bolt hasn't get there yet. And for the potassium, are the potassium channels open or closed in this section? Potassium, the blue ones, are they open or are they closed?
They are close.
They are closed. Very good. Let's take a look at here. They don't open until +30, right? We are nowhere near +30. We're still in the negative zone, so they are going to be closed. So that's what we're going to draw. Again, I'm leaving the sodium potassium pump out. They are still there, but they're not going to be having any significant action on what we are talking about. Okay. Now, potassium has nowhere to go. They are still stuck. Same thing here. Okay, you have some outside, just a few dots here and there. And we can say high potassium inside, low potassium outside. Same thing over here. And for the sodium, because the doors are open now, they will come in. They really want to come in. Right? They really want to come in. All going to come in. Nothing's going to happen here because the doors are still closed. So the sodium is still high on the, oops, it's still low on the inside. Still low over here. Just like before. Oh my goodness, what am I doing? They are still low on the inside. Okay. Low on the inside and high on the outside. Okay, just take a look at this for a moment. On the right side is exactly the same as before. Nothing has changed because no doors are open. This is exactly the same. Over here, the only thing that's different from before is that the sodium channels are now open. Once you open the sodium channels, all those sodiums are going to be coming in. Right? Remember I said, let's say Black Friday sale, right? They all want to come in. Okay. As a result of this, afterwards, you will have on the inside, you would have high sodium. And then outside we'll have low sodium. Okay, I'm going to give you a moment to catch up. Let me know when you're ready, and then I'll tell you more. So looking at the graph, which is next to you, it was flat, and then we crossed the threshold. What's going to happen next? Okay, so over here at the threshold potential, we understand the sodium channels open. And then the sodium is going to be coming in. As the sodium comes into the cell, what will happen to your membrane potential? Is it going to go up? Is it going to go sideways? Is it going to go down? Let's do a vote. Okay, we have the sodium coming in. What is going to happen to the graph? What is going to happen to your membrane potential with all the sodium coming in?
A/1, please.
Oh, can I uh explain again? Is that what you're saying? The
Yeah,
question? Yeah.
on the pole is the A, B, C.
Oh
But
yeah, 1, 2, 3 is A, B, C. Yeah. Is it A, B, or C? Okay, you have the sodium coming in now. Is that gonna make your membrane more positive, no change, or more negative? That's the question. Okay, well, I know it's really complicated. I'm sorry. Let's go back to here and take a look. Okay, remember it was negative on the inside to begin with, but now with all the sodium coming in, sodium carries a positive charge, right? So all these extra sodium coming in is gonna make the inside more positive, isn't it? Right? So it's gonna go up. It's gonna go up. Positive, 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 positive. Oh, sorry guys, I gotta restart again. Hmm. Okay. It's gonna go up. But it's not gonna go up forever. It's gonna go up, it's gonna go up, 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 until what number? What number do we stop going up? Well, the sodium is gonna come in as long as the channels are open. But if you take a look at here, the sodium channels will close at positive 30, which means as the sodium are coming in and changing the membrane potential, eventually it is going to reach the closing voltage for sodium channels. And once you close the sodium channels, then the sodium stops coming in. Right? So this portion here is caused by sodium coming in. We call that sodium influx. And it causes your membrane potential to switch from negative to
To
positive.
positive.
That's right. So we call this depolarization. Depolarization. To polarize in this context means to make it more negative. To depolarize is to make it more positive. Okay, so the rising phase of the graph is called depolarization, and it's caused by the sodium coming into the cell, or as we call it, the sodium influx. Now, on our drawings, basically what happens is it's just a little tipping point, right? Once you go over negative 60, Then you open the doors and all the sodium comes in. It's going to push you all the way to positive 30 millivolts. Okay, so at this moment, the inside will now be positive, as shown by the graph, and the outside will be negative. Now, further down, it's a different story. Further down the axon, we haven't have the lightning bolt yet. So it's still negative on the inside and then positive on the outside. Are you guys following the story so far? Any questions? We are almost there.
Sir, please.
Yes.
I don't know if I follow very well. Initially, the inside of the membrane was 70, minus 70.
Yeah.
What did happen? And it goes from minus 70 to minus 60.
Right. So we haven't talked about that portion
Okay.
yet, right? Yeah. And remember I said just trust me for now that it will when a trigger arrives, it's gonna push it from negative 70 to negative 60. And once you cross the negative 60, it triggers all these events that are happening. And in a moment, we're gonna come back and explain how it goes from negative 70 to negative 60. And in fact, right now, we're gonna get there very soon. Okay, so we are at the top of our graph, which is right here at positive 30, roughly positive 30. And three things are gonna happen at this point. And three things gonna happen. Let's write it down. Number one. Oh my goodness, what's going on? Ugh, I'm so sorry. Gotta restart this again. I gotta talk to IT people. Maybe they'll fix it for me. All right. Do you guys use this thing in math as well, or like is there like some other program that they use? I'm curious. It happens in math too, yeah? I think we are all having the same problem. Okay, number one, what's going on here? First thing that's gonna happen is the sodium channels. Sodium channels will close. Okay. It's only gonna open between negative 60 to positive 30, right? It's gonna close and it's gonna enter the refractory period. You gotta wait for the bath, the toilet to fill up again before you can activate it. You gotta wait for the channels to reset before they will react again. So you close the channels for the sodium. Now, if you pay attention to the note we roll, and boy, we roll a lot of notes, the potassium channels has an opening voltage of 30 millivolts. Right, so once you reach 30, you shut off the sodium, but now it's time to open the potassium. It's time to open the potassium. So that's number two. Potassium channels open. Okay? Tell me, please tell me what's going to happen to the potassium. Where will they go? Absolutely, they will go out, right? Look, think of, again, the Black Friday analogy. All these blue Walmart employees that have been trapped in the store preparing for this day for so long. They really want to get out, but it's not the end of their shift yet. And now, finally, it is. The doors are open and they will exit, right? Once all the blue doors are open, they will exit. So let's draw one more picture. This is the last one we're going to draw. I know it's very messy. So, take the time to reorganize it if you need to. Okay, so this next part is called repolarization. Repolarization. Okay, so what's going on here? Let me just set the stage for you. We have the sodium channels, and we're going to close them now because we are sitting at positive 30. All right, that's the closing voltage. Okay, we're not going to draw the further down stuff yet. Okay, leave it alone. Going to add to that later. And so all these sodium that came in, they are done. Okay, they have nowhere else to go. They cannot go back out. Doors are closed. Too bad. This is the high sodium right now and low sodium, relatively speaking, of course. And then it is time we pay attention to the potassium. There we go. And all the potassium that were trapped on the inside, they really want to leave. They really want to leave. Okay. They are so done. Gonna leave like that. Okay. Now here is a important thing to note about the potassium. There are two driving forces that push the potassium ions out of the cell. Okay. There are two driving forces that would push them out. Number one, concentration gradient. Okay, that part is not hard to understand, right? Like it was high on the inside and low on the outside, so obviously they want to leave. Okay, things naturally wants to go from high to low concentration, so that's gonna push them out. But there is a second driving force that would really push the potassium out, that would really encourage them to go out, and that is electrostatic repulsion. What does that mean? Well, potassium is positively charged, and so is sodium. With all the sodium coming in from the previous action, the inside is really positive, right? You can see with the pink symbols here, it's really positive, and positives don't like to be next to each other, right? So the potassium will try to get away from all these positives that are coming in, and the only way the only place to go is to leave the cell. Okay. Now you might be thinking, how come the sodium don't want to get away? They do, but they can't because the doors for them are closed, right? So the only thing that can move and get away from all these positive charges would be the potassium. And the only place they can go would be out of the cell. So down its concentration gradient, the electrostatic repulsion will cause mass exodus of these potassium. So I want you to think again with me, what's going to happen to your charge when all the potassium are leaving the cell? Is the membrane potential going to go up further? Is it going to be sideways or is it going to be coming down? What's it going to be this time? How are we going to finish our graph here? Right. That's correct, Elphys. And I hope the rest of you also understand, right? It's going to be coming down. And it's going to be coming down hard, okay? Really, really, really come down. Because like I said, there are two driving forces that push the potassium out, right? So we're going to reestablish the negative charge which is called repolarization. And the main cause of this is potassium efflux. Okay, influx is coming in, efflux is leaving. Okay, so repolarization, all coming in. And it's going to take you all the way to -90. -90. Around -90. Okay. Now let's put some more markers here to help you, I don't know, connect the situations. At the beginning, we had low sodium, high potassium because of the sodium potassium pump. But once you open the sodium gate, all the sodium comes in, and at +30, the inside will have high sodium because they just came in, and also high potassium because they didn't do anything yet. Okay, so that's why the inside is like super positive right now. And you will close the sodium channel, you will open the potassium channels, then you have mass efflux of the potassium. So when it's all said and done, you will have low potassium, but you will still have high sodium because, once they came in, they have nowhere to go, right? They are trapped. The doors are closed for the sodium. Okay. So at this point, we have two situations. Number one, we are more negative than resting potential. We're too negative, in fact. So this is called hyperpolarization. Again, if you're confused with all these words, polarization just means negative. Right? You can think of it like that. In the context of what we're talking about, polarization means negative. Okay? So when you are depolarization, you're making it less negative. Repolarization, you are recreating the negative charge. And then hyperpolarization means you're too negative. Our reference is, of course, the resting, right? You're too negative. So at this point, there are two problems. The membrane potential is too negative. Right? We cannot get the neuron to fire again unless it's at -60, right? So -90 is like, way below that. And then second problem is the ion distribution is opposite compared to resting potential. Okay, so two things. Compared to the beginning, number one, we are too negative. Number two, look at the ions. It was supposed to be low sodium and high potassium, and now it's backwards. Okay? So we gotta fix this. We gotta fix it. We gotta bring it back up to -70. We gotta redistribute the ions so that it's back to normal, and then we gotta fix the membrane potential so it goes back to normal as well. What is going to do that for us? Okay, of all the things we wrote down in the notes, what is going to come and fix this problem for us? Because if we don't get back to -70, if we don't redistribute the ions, the neuron will not fire again. So we need something to take the excess sodium out and bring back more potassium. So what's going to do that? Well, it is going to be exactly what you said, right, Elvis? It's going to be the sodium potassium pump will reestablish. Is that even a word? Reestablish. Recreate. Yeah, you know what? Let's just say recreate. Probably easier. recreate the resting potential conditions. Okay? Almost there. Almost there. Okay. Remember, I said there are three things that's going to be happening over here. I'm going to talk about it in a second. Please catch up with all the writings. Okay, any questions? We'll be done soon, okay? Maybe 15 more minutes and I'll let you go. Hang in there, guys. So let's go back to the other drawing and and fill in some of the gaps that we might have. Over here, um we um we know the sodium all came in, and then as the potassium are leaving, the sodium is going to be high over here. They cannot go back out, right? I told you already. The sodium channels are closed. But they don't want to hang around here either. It's very positive, right? With all the potassium here, it's like it doesn't want to be there. The repulsion, right? It's very strong. So take a look at this picture and tell me, once the sodium comes in and you close the door, there's only one place it can go to get away from all these positive ions. Okay, remember, they cannot go back out because the door will close soon. There's only one place they can go. Can you see where it is? There's only one place it can go. Okay, let me zoom in a little bit for you. Okay, remember, these doors will close momentarily. All these sodium coming in. Where else can they go? Do you see it on the picture? Okay, yeah, sure. Some of it might get pumped back out with the pump, but not yet. Not yet. Okay. Where is it gonna go? Okay, just look at the picture. Okay. Things will go from high concentration to low concentration. They can't go back out because the doors will be closed. Where else is it going to go? That's right. Okay, someone gave me the answer. To the right, because further down the axon, the sodium is still low because nothing has happened yet, right? So some of these will creep forward. It's like, hey, it's not a lot of sodium further down. Let's go there. And listen to me carefully. As the sodium moves further down the axon, they will bring the positive charge with them. So this number, -70, is gonna become -65, -62, -61, and eventually, if enough sodium moves forward, it is going to become -60. And that is going to be the triggering membrane potential, right? The threshold potential for the sodium channels. And then all the sodium channels here will now open and they will go through the exact same thing that had just happened for the previous section, right? And all these sodium is going to come in, and then some of it will go forward, And then it will push the next section over the threshold, and then the next section, and then the next section. Do you understand? Right? So what's going to happen is here, as the potassium leaves, instead of positive on the inside now, it will go all the way down to negative 90, as I told you. But then the next section will be pushed over the threshold, and then it will go all the way to positive 30 because the sodium channels will now be open. I'm not going to draw the potassium. They don't play a role at this point. And then the sodium will come in, right? So then, I know it's getting very messy. As this becomes negative again, because of the potassium leaving, the next portion is going to become positive now, just like it did previously. Do you see that? I will try to explain this one more time as a whole process in a bit. Okay? But copy what you can, and then we'll go from there. Okay, this is like the most confusing part of the nervous system. Once you get past this part, you'll be okay. Okay. This is almost worse than it is worse than, I would think it's worse than the immune system stuff that we talked about. But it is what it is. Okay, so on our graph here, I am gonna add one more thing for you. Once you reach positive 30, the sodium channel closes, the potassium channel is open, and number 3, sodium channels further down the axon opens. Okay, the ones of the current section will close, right? But some of the sodium is going to creep forward, and it's going to cause them to open, right? So that's kind of how it went from negative 70 to negative 60. You have to wait for the sodium to kind of creep forward to push it over the threshold. It's exactly like dominoes. Okay, exactly like dominoes. You push this one, and then you push the next one, and push the next one, push the next one. Okay, but until the trigger gets there, you will be standing. Okay. So where is the action potential? Well, the action potential is referring to this portion of the graph. Okay, this portion of the graph. This portion of the graph is the action potential. Action potential. Which by definition is the temporary reversal of membrane potential. Okay, you are flipping it from negative to positive only momentarily before it goes back. Right? So like the flipping to positive will cause the next sections to flip, right? And then this one's gonna get reset, and then it will cause the next one to flip, and then the next one to flip, and the next one to flip. Okay. you know, you see people doing this at the concert, right? You know, the wave, like so, like you stand up and then you sit down, and then it causes the person next to you to stand up and then sit down, right? So it's like that, right? It will cause the flip, and then the next one will flip, but you don't stay standing up. Once you get up, you gotta sit down again. So once you flip to positive, you gotta flip back to negative, right? So positive, and then you cause the next one to become positive, and so on and so forth. Okay, so that's the graph. That's the whole graph. And it's an important graph. I'll post it on the notes. You should reorganize it, add some more details if you want. If you understand this graph, you should be okay for the quiz number 2, which you don't have to worry about for now. Okay, quiz number 2 is not going to happen until after the test. Okay, which it's on February 19th. Okay, don't worry about the test for now first. All right, so let me try my oh, sorry, I forgot to mention something here. Like that threshold potential, sorry, the refractory period, like where does it come into play for all this? So listen, um, negative 60 is the opening threshold for the sodium channels. So you're gonna open them at negative 60. Sodium comes in, you're gonna push it to 30, then it closes. Once it closes, it enters the refractory period. It's not going to respond until it resets itself. So on the way down to our graph, even though you reach negative 60 again over here, right? This does not trigger sodium channels. Again, because they are in refractory period. Because otherwise, if they are not in refractory period and you open them again, then the sodium will just goes back in, and then it just fluctuates up and down. It's like it's not going to be a meaningful uh, signal. You're not going to be able to move the signal forward. In other words, okay. So by having the refractory period, it ensures the signal will go only in one direction, and because we need the sodiums to move forward, right, to trigger the event in the next section. You cannot open the sodium channels again, and they might leave instead of going forward. All right, I'm gonna try to explain this one more time in a different way, and then I will let you go. Here we go. I think we're doing quite good with time, actually. So you don't have to draw this. You just sit back and watch. Okay. Yeah. So someone just asked for the test. Yes, the test will be extended. The due date, the new due date for the test will be Sunday, 11:59 p.m. Okay, that's the new due date. New due date. Test 1. Okay, but you will have access to it on Friday. I'll send an email out. Okay. All right. So this is the axon. This is the neuron. Okay. Don't draw this. Just let me explain. Okay. This is the neuron. Okay. And then it's gonna be with the axon terminal right there. And I am going to copy a bunch of these. So that's why you don't have to draw your own. You just watch. Okay, and hopefully it will make sense. So some kind of trigger arrives. Okay. Like there are green signals and red signals. If there's more green signals, then we're gonna have a trigger, a stimulus. I still haven't really explained to you. Like you understand what happens along the way, but I haven't tell you what happens at the beginning, like where does this come from and what happens at the end. That's next class. So, the trigger will come, right? Actually, you know what? Before the trigger, let's just keep all the numbers like this. All of them are negative 70, negative 70, negative 70, negative 70, okay? And then the trigger will come. No, I shouldn't have erased that. Stimulus will come. And what's going to happen? Well, what's going to happen is you're going to push this over the threshold, negative 60. And then the sodium will come in. And as the sodium comes in, it will reach 30, positive 30. Okay, so this section is basically going to light up. Okay, positive, lights up. The rest of it will still be at negative 70, negative 70, negative 70. Then what happens is, because this part is undergoing the action potential, the flip, right, all the sodium coming in, and some sodium will creep forward because that's the only place they could go. It will then cause the next section to go over the threshold, and then the sodium will come in for the next section, causing it to become positive 30. However, for the previous section, after all the sodium comes in, you close the sodium channels, you open the potassium channels, and then the potassium will start leaving the cell, right? The efflux. And that will bring this all the way down to negative, negative 90. Negative 90. Right? So this is negative 70, negative 70, negative 70. Right? And then what's going to happen next? Well, what's going to happen next is this, all the sodium coming in for this section, some of it will creep forward, and then it will cause the next section to go over negative 60. And then it will become positive 30 because of the sodium coming in for that section. And then for this section, it will plummet all the way down to negative 90 because of all the potassium leaving. What about that? Well, once you get to negative 90, the hyperpolarization, way too negative, the sodium potassium pump is going to help reset it for you, and it's going to be back to negative 70. Okay? And then the rest of the story, you know, right? The positive 30 is going to cause some sodium will creep forward, and then it will cause the next section to become negative 60. And so it will flip the next section and the next section and the next section until you get all the way to the end, the axon terminal. Okay, so as you are flipping the signal forward, you are resetting the signals behind. You're resetting the membrane potential back to negative 70. Right? So if you look at this and in its entirety, you know, only one section is flipping, and the flipping is not permanent. It's a temporary flip. And that is the definition of an action potential, the temporary reversal of membrane potential. And that's how the signal gets propagated down the axon. Okay, think of it as like a light bulb, right? Like, you know, lights up, ding, and then it causes the next section to lights up, and then lights up, lights up, lights up, lights up. That is all I have. Anything you want to ask? Anything you want to tell me? Questions about the test before I let you go? Okay, well, you know where to find me. If you have questions, just email me. Good luck with math, chemistry, and my tests, and I will see you all on Monday again. Okay, take care, guys. Bye-bye.
Bye, sir. Thank you.
Yep.